Welcome to Biochemistry Simplified with Dr. Onas, and let's simplify biochemistry.
Hi, Biochemistry Simplified. In today's video, we are going to be talking about the major pathways of carbohydrates. Before we get into this video, please continue to support us by hitting the like button, commenting down in the comment section, and please subscribe. Alright, let's get into it. So, what are carbohydrates? Carbohydrates are the major source of energy of the living cell, and carbohydrates are also known as saccharide. They are also known as saccharide, and the word saccharide comes from the Greek word saccharon, meaning a sugar. Carbohydrates are the most abundant molecule on Earth by mass, and much of carbohydrates are produced by photosynthetic organisms such as bacteria, algae, and plants. An example of a bacteria that can photosynthesize is cyanobacteria. So, these photosynthetic organisms are capable of converting solar energy to chemical energy, which are used to make carbohydrates on carbon dioxide. So, the example of carbohydrates, we have rice, carrots, millet, bread, banana, and so on. So, the simplest form of carbohydrates is monosaccharide. Monosaccharide comprises of glucose, fructose, and galactose. So, these are the Fischer projection of glucose, fructose, and galactose. So, this is glucose. They are all from the family of azoles. That is, they all contain 6 carbon. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5. Alright. So looking at glucose and fructose, they are different. How? At carbon 1, glucose has CHO, that is, as its functional group, that is, it belongs to an aldehyde group. While fructose at carbon 2 has C double O, that is alkanol or a ketone. So fructose belongs to an alkanol or a ketone group. All right. And looking at the structure from carbon 2, so OH, but at carbon 3, it changes, right? So, for glucose, there is change in conformation at carbon 3. All right, and this carbon 5, you can see that this OH is at the right. So, that makes it D-glucose. D means dextrorotatory. That is, it rotates to the right. So that makes it D-glucose. If this OH rotates to the left, it will be L-glucose. The same thing applies here, and the same thing applies here. So if the OH rotates to the left, it will be L-glucose. That is levorotatory. But since it is not on the right, it is only dextrorotatory. All right. So let's move on. So you can see that looking at this, D-glucose is similar to D-galactose. For the glucose, it's only at carbon 3, while for the galactose is both carbon 3 and carbon 4. There is change in the configuration in carbon 3 and carbon 4, while for glucose only at carbon 3. So fructose also, there's change in configuration at carbon 3. But this carbon 2 is double O, that is, so it is a ketone. While these are aldose. This is a ketose, these are aldose. You understand that? Alright, so now let's move on to the major pathways of carbohydrates. The first one is glycolysis. So glycolysis is a pathway that involves the oxidation of glucose into 2-monopyruvate and 2-monolactate. So it converts glucose to 2 mode of pyruvate and two mode of lactase. Hope you understand that. So looking at the citric acid cycle, so the next pathway is citric acid cycle. Citric acid cycle is also known as Krebs cycle and also known as tricarboxylic acid cycle. So this particular pathway involves the conversion of acetyl-CoA. The conversion of acetyl-CoA to carbon 4-oxide or carbon dioxide. And it is the final common pathway. This Krebs cycle or citric acid cycle is the final common oxidative pathway for carbohydrates or amino acid or fats through acetyl-CoA. That is, acetyl-CoA can link to carbohydrates, can link to amino acid, can link to fats. So moving on to the next pathway, which is glucogenesis. Glucogenesis involves the formation of glucose from non-carbohydrate precursor. It involves what? The formation of glucose from non-carbohydrate precursor. That is, we are synthesizing glucose from a precursor that is not carbohydrate. For example, we have amino acid, we have glycerol. So moving on to the next pathway, which is glycogenesis. Glycogenesis is the formation of glucose from a glycogen. So moving on to the next pathway, we have glycogenolysis. Glycogenolysis, which means what? Which means the breaking down of glycogen back to glucose. So glycogenolysis is what? Is the opposite of what? Of glycogenesis. Do you understand that? So moving on to the next pathway, we have azote monophosphate shunt, which is known by its acronym HMP shunt. HMP shunt. And it's also known as Pentose phosphate pathway, PPP. So this particular pathway is an alternative oxidative pathway of glycolysis and citric acid cycle. That is, it involves the conversion of glucose into carbon 4-oxide and water. So the next pathway is uronic acid pathway. This particular pathway converts glucose to gluconic acid, pentose, and ascorbic acid in animal, but not in man. Hope you understand that. And moving on, we have galactose metabolism. Galactose metabolism pathway. So this galactose metabolism pathway can be converted back to glucose and it can be used to synthesize lactose. So looking at it, so this is galactose. So this galactose can be used to what? To form glucose. And this same galactose can be used to synthesize lactose. That is, glucose and galactose will give us lactose. Hope you understand that. So lactose is a disaccharide. So glucose plus glucose will give us maltose. And glucose plus fructose will give us sucrose. So maltose, sucrose, and lactose are examples of what? Of a disaccharide. Then moving on, we have the fructose metabolism. This fructose metabolism involves the conversion of fructose into pyruvates. And also, there's a relation between fructose and glucose metabolism. So look at it. This is fructose. This is fructose metabolism pathway. It starts with fructose. Fructose can be converted into fructose 1-phosphate by the enzyme fructokinase. Hope you understand that. The 1-phosphate can cleave into dihydroxyacetone phosphate and glyceraldehyde by the enzyme aldolase D. Here is the enzyme. So Both dihydroxy acetylphosphate and glyceride can now go further to produce pyruvate. All right, hope you understand that. So the last one is the amino sugar and mucopolysaccharide metabolism. So this particular pathways involve the synthesis, like we are synthesizing amino sugars and other sugars at first. Hope you understand that. And after synthesizing amino sugars, we now use the amino sugars and other sugars to form for the formation of what? For the formation of mucopolysaccharide. But the question is, what are amino sugars? Amino sugars are monosaccharide, which hydroxyl group is changed to amino group. Hope you understand that. Amino sugars are monosaccharide, which hydroxyl group is changed to amino group. So look at it. Amino sugar is what? Is a monosaccharide. Can be glucose, can be fructose, can be galactose. So if now decide to change one of the hydroxyl group to a amino group, then we are going to get amino sugar. We are going to get amino sugar. We are going to get amino sugar. An example of amino sugar is glycosamine, galactosamine, and so on. So if we change the OH of a glucose, one OH of a glucose to NH2, then we're going to get, instead of glucose, now be glucosamine. And then we change one OH of galactose to NH2 to be galactosamine. Hope you understand that. So as I said, that the amino sugar and mucopolysaccharide metabolism, so these pathways use amino sugars and other sugars to form for the formation of what? For the formation of mucopolysaccharide. So what are mucopolysaccharide? Mucopolysaccharide are also known as glycosaminoglycan, GAGs, GAGs. So these mucopolysaccharide examples are high hyaluronic acid, creatine sulfate, heparan sulfate, chondroitin sulfate, dimethyl sulfate, and heparin. Hope you understand that. All right, so that is all about the video pathways of carbohydrates. So before we end the video, so let us look at those that are oxidative and those that are synthetic. So any pathway that has lysis as its suffix, it means that such pathway is undergoing oxidation. That is, such pathway is what is breaking down. So for this pathway, we have glycolysis, we have citric acid cycle, we have Glycogenolysis, we have EPP, Pentose phosphate pathway, which is also Pentose monophosphate shunt. So for the synthetic pathway, we have Gluconeogenesis, Glycogenesis, an example of oxidative pathway is uronic acid pathway, because it also involves the oxidation of glucose to gluconic acid, phosphates, and ascorbic acid in animal, but not in man. So whenever you see that a pathway has genesis as its suffix, such pathways are undergoing, or it is forming or synthesizing. Do you understand that? So such pathway, it is a synthetic pathway. All right, so that marks the end of this video.